What is the question? Vitamin which is needed for the process of transamination. See, transamination is the process where amino acid transfer its amino group to keto acid. Okay, when that happens, the amino acid is converted to respective keto acid, and the keto acid which is going to accept the amino group is going to get converted to amino acid. So this is the transamination. Transamination means transfer of amino group from one amino acid to one keto acid. This is catalyzed by transaminase enzyme. This enzyme is needing PLP. PLP is pyridoxal phosphate. What is pyridoxine? Pyridoxine is vitamin B6. So pyridoxal phosphate is active form of vitamin B6, right? So the PLP is required. Without it, this transamination will not be occurring. So this is very first step of catabolism of various amino acids, where the amino group is safely transferred to keto acid. And most of the time, if you want to know little more, most of the time this alpha keto acid which is going to accept the amino group is alpha ketoglutarate, which is intermediate of TCA cycle. So once the alpha ketoglutarate accepts the amino group, that is converted to glutamate. Okay, that is converted to glutamate. So this is transamination, and as the question is asking you which vitamin is required, answer is C, vitamin B6 is required. Now coming to second question, which is talking to you that one double-stranded RNA genome is having the uracil 15%. So if uracil is 15%, then adenine also will be 15%, because uracil pairs with adenine in double-stranded RNA. So this is 15% and this is 15. Collectively it is 30%. Now the question is, what is the percentage of guanine in this genome? See, guanine pairs with cytosine. So if 30% of total is contributed by uracil and adenine, the remaining 70% will be contributed by guanine and cytosine, which will be equally in percentage, 35 and 35%. So this question is asking you, what is the percentage of guanine in this double-stranded RNA? The answer is going to be 35%. That is, answer is C, 35%. So this is how you have to calculate what may be the percentage of one particular specific nucleotide or corresponding base if the percentage of any specific nucleotide or the base is already given to you. Just by remembering the simple concept that if it is DNA, the A is going to pair with T, C is going to pair with G, and if it is RNA, U is going to pair with A and C is going to pair with G, right? So this is a little mathematical question which is very, very often asked to you. Very repeatedly it's being asked. Now coming to the third question, before being oxidized, fatty acids are activated in cytosol to form which of the following? The answer is going to be acyl-CoE. See, when we are talking about the fatty acid beta oxidation, we all know it is a mitochondrial process. All the enzymes of beta oxidation are existing in the mitochondrial matrix in the dispersed fashion. They are not making any complex, rather individual enzymes are there in the dispersed fashion. It's not like fatty acid synthesis where enzymes are present in a complex pattern where they all are present at one particular huge complex that's called fatty acid synthase complex. There the enzymes are collectively present in the cytosol in the form of fatty acid synthase complex. But when we are talking about beta oxidation, all the enzymes are individual, they are dispersed fashion. So when you're talking about the fatty acid beta oxidation, this fatty acid need to enter the mitochondrial matrix. But one thing you should understand, that free fatty acid can never enter the mitochondrial matrix because it is not permeable to outer as well as inner membrane of mitochondria. So what happens, first of all, this fatty acid is converted to acyl-CoE by a specific arrangement at the outer membrane of mitochondria, which we call it as acyl-CoE synthetase, right? Acyl-CoE synthetase. This is part and parcel of carnitine shuttle. So acyl-CoE synthetase, what it does with the expenditure of ATP, which is converted to AMP, that calculate for 2 high energy phosphate expenditure. So with the expenditure of 2 high energy phosphate, the fatty acid is converted to acyl-CoE, and once the acyl-CoE is formed, rest of the components of carnitine shuttle are able to carry this acyl-CoE to the mitochondrial matrix. So in the cytosol, this kind of activation of fatty acid is taking place, or the conversion of fatty acid to acyl-CoE is taking place, in which we have expenditure of 1 ATP, but actually there are 2 high energy phosphate which are spent in this process. So the question is very simple. This is one of the component of carnitine shuttle which we have discussed. Alright, now coming to question number 4. Now, which of the following is a feature of non-competitive inhibition? Be very careful. If these competitive and non-competitive questions are being asked, you have to know little basic points. For competitive inhibitors, you remember that Vmax is same, but Km is increased. The apparent Vmax, that is the Vmax which is present in presence of inhibitor, is still going to be the same in presence of competitive inhibitor, but to get the half of that Vmax velocity, the amount of substrate desired is going to increase. That's competitive inhibitors. When we say non-competitive inhibitors, the specific characteristic of non-competitive inhibitor is that they act in such a manner that the apparent Vmax is going to decrease. Apparent Vmax is going to decrease. Apparent Vmax is going to decrease. But to get the half of that new load velocity, the amount of substrate is going to be the same. So Km remains same. The apparent Km, we call it as apparent Km. The new Km which we talk in presence of inhibitors, we call it as apparent Km. We represent it by apostrophe. Similarly, the new velocity which we are discussing in presence of inhibitors, we call it as apparent velocity, that we represent it by apostrophe, V apostrophe max. If we just write Vmax, means we are talking about the velocity maximum which is present in which is seen in presence of enzyme, pure enzyme, without presence of any inhibitor. But when we are talking about the new velocity and new Km which we derive, whether it is same or it is different, we call it as apparent Vmax or apparent Km. So that this type of questions are repeatedly asked. They will not stop asking this question, that what happens to Vmax, what happens to Km in competitive inhibitor and non-competitive inhibitor. So this is don't go into mechanistic detail that how it happens, why it happens, or why not it happens. That's not going to help you at all. You just remember these calculations, that when we are talking about competitive inhibitors, the apparent velocity will reach, but at a very large amount of substrate, if it is being present, then only the apparent velocity will be same as the original velocity. But still, we can still say that, okay, at one point of time, the apparent velocity will be same as that of original velocity. And Km, as I have told you, it's going to increase, and in non-competitive, the velocity maximum is going to decrease, because non-competitive inhibitors, they destroy the active site. They doesn't allow the enzyme to act in the original fashion, and they hence are going to reduce the velocity maximum. But mathematically, it is seen that to get that new load velocity, to get half of that new load velocity, the amount of substrate desired is going to be the same. Now coming to question number 5, pancreatic amylase. I've seen this question, very easy question, but students, they make blunder in this question. This question is talking about the digestive enzyme, which is pancreatic amylase. Pancreatic amylase is a digestive enzyme which acts on the starch and glycogen and breaks it into a simpler form, that is disaccharide, trisaccharide, oligosaccharide. If I talk to you about the glycogen or the starch, they have the branched polymer of glucose. I have drawn one portion of our glycogen, say for example, where these round units are the glucose, and the bonds which are connecting the glucose together are glycosidic linkage. You have to be very clear that this bond is alpha 1,6 glycosidic linkage, which are linking the glucose together. And further on this branch, when we go to the external end, we have the bond which is 1,4 glycosidic linkages. So these all are 1,4 glycosidic linkages. If you're talking about the pancreatic amylase, this is going to digest this glycogen or starch into simpler form. And please understand that this amylase is not going to act on this bond, that is external most 1,4. Rather, the characteristic of pancreatic amylase is that it's going to act internally. Internally, remember this word, internally means the internal, not the external most 1,4 bond, but the internal 1,4 bond is being cut like this. So suppose if this bond is cut, you tend to have 2 glucose together. I mean, disaccharide you're going to get. Suppose little more interior bond is cut, you tend to have trisaccharide. So either you tend to have a disaccharide or trisaccharide, you never tend to have a monosaccharide by action of pancreatic amylase. That you should remember. Now I said this question is a very easy question, but the student make a blunder. What students say that pancreatic, if I ask this question, they directly jump into the answer that external most bond is cut or the exterior 1,4 is cut. It is not the case. This exterior bond is cut by natural enzymes which is found in our cell, not GIT, cell, and that is glycogen phosphorylase enzyme. बच्चों, glycogen phosphorylase enzyme cuts the external most glycosidic bond, releasing 1 glucose at a time. So this is the characteristic of glycogen phosphorylase enzyme. So try to differentiate the action of glycogen phosphorylase and pancreatic amylase enzyme. They have got their specific point of their action. So when I talked about question number 5, the pancreatic amylase is going to act on internal 1,4 glycosidic bond, right? Internal 1,4 glycosidic bond. It doesn't act on 1,6 bond. It acts only on 1,4 bond, and that too internal glycosidic bond, 1,4 glycosidic bond. Now coming to next question. Alpha-linolic acid is considered to be nutritionally essential. Yes, we cannot synthesize this alpha-linolic acid, but this is certainly important for us because it is involved in prostanoid synthesis, prostaglandins and eicosanoid synthesis, various other eicosanoid synthesis. So this is nutritionally essential because human cell cannot synthesize this alpha-linolic acid. So that need to be provided in diet. Now the question is asking why it is considered nutritionally essential. Why human cell cannot synthesize this alpha-linolic acid. For that you should know this is 18 carbon and having 3 double bond. We have oleic, linolic and alpha-linolenic. Remember this in this particular order. Oleic, linolic and alpha-linolenic. Oleic is having 18 carbon 1 double bond, linolic is having 18 carbon 2 double bond, and alpha-linolenic acid is having 18 carbon and 3 double bond, and the position of 3 double bond are 9, 12 and 15. We have desaturase enzyme which introduces the double bond at various positions like delta 9 position, delta 6 position, delta 5 position, delta 7 position. In fact, we have all enzymes. We have different enzymes, desaturase enzyme, which can introduce the double bond in human fatty acid chain till delta 9 position, not beyond that. The 12 and 15 position double bond cannot be introduced by so-called desaturase enzyme, which is characteristically found in endoplasmic reticulum. So when these double bonds are not being introduced in the fatty acid chain, which is freshly synthesized on fast complex in cytosol, obviously the alpha-linolenic acid cannot be produced in human cell by its metabolism. So this alpha-linolenic acid need to be supplied in the diet. So this is the funda why this alpha-linolenic acid is the essential fatty acid. Now the question is asking you why it is essential fatty acid. Let's see the answer. Now the answer is very clearly written in this one of the option that the human double bond cannot be introduced into the fatty acid beyond delta 9 position. We cannot introduce the double bond at delta 10, 11, 12, 13, 14, 15 position. So those fatty acids which are important for our metabolism, but they have the double bond beyond delta 9, they need to be provided in diet. Even linolic acid, as I've told you, it's having 18 carbon 2 double bond. Linoleic acid. This is also involved in icosanoid synthesis, and this 2 double bond is present at delta 9 and delta 12 position. Delta 9 can be introduced, but delta 12 cannot be introduced. So again, linoleic also becomes an essential fatty acid. So we have 2 essential fatty acid, linoleic and alpha linolenic acid. Arachidonic acid is semi-essential fatty acid because once provided, the linoleic can get converted to arachidonic acid. So arachidonic acid is said to be semi-essential fatty acid. It is not purely essential fatty acid. Now coming to the next question, that is question number 7. ACAT is activated by. ACAT is an enzyme which is intracellular enzyme. This is activated by free cholesterol in the cell. Like if I draw, this is one of this extrahepatic cell, and this is having the cholesterol pool where the cholesterol is stored in the free form. We have enzyme called ACAT enzyme. So first question is, point is that ACAT is an intracellular enzyme, and this ACAT is acyl-CoA acyl transferase enzyme. Acyl-CoA cholesterol acyl transferase enzyme. The complete name is acyl-CoA cholesterol acyl transferase enzyme. So this enzyme transfers the fatty acid to the cholesterol as to convert it to cholesterol ester, which is the storage form of cholesterol in the cell. If you attach a fatty acid to the 3rd hydroxyl of the cholesterol, it becomes a cholesterol ester, which becomes an inert form of cholesterol, which becomes a storage form of cholesterol. So this cholesterol is stored in the cell. So whenever we have excess amount of free cholesterol in the cholesterol pool, what happens? This enzyme undergoes activation. So kind of like cholesterol is sending the signal that I'm excessive and convert me to the cholesterol ester, which is the storage form. So the matter is very simple, logic is very simple, that excessive cholesterol in the cell pool is the factor which is going to activate the ACAT enzyme, which then is going to act and transfer one acyl chain to the cholesterol to convert it to cholesterol ester, which becomes a storage form of the cholesterol. Don't confuse this name of the enzyme ACAT with ALCAT. ALCAT is altogether a different enzyme which is circulating free form in the plasma, and this ALCAT is activated by ApoE1 of HDL, and this ALCAT is involved in reverse cholesterol transport. Reverse cholesterol transport, for which HDL is required. Reverse cholesterol transport. So we have ALCAT enzyme, we have ACAT enzyme. So that is how they are going to act. Now coming to question number 8. We all know that tag is stored in adipose cell. Now this question is asking you, all of the following are true regarding this tag storage in adipose cell, except, let me tell you, the answer is going to be glycerol kinase factor. Glycerol kinase is the enzyme which is not found in adipose cell. So it doesn't play any role in storage of triacylglycerol in adipose cell. Remaining all other statements are all right. To explain this, I will go in brief detail of the concerned concept. Suppose this is adipose cell, and you are waiting that how the tag is formed and it's stored. See, for the formation of tag, triacylglycerol, we need to have two material. One is the glycerol 3-phosphate, on which the acyl-CoA is getting esterified. 3 molecule of acyl-CoA is getting esterified, and when this happens, we tend to have the formation of triacylglycerol. The major question is, what is the source of glycerol 3-phosphate in adipose cell? The answer is glycolysis. The glycolysis, where the glucose is broken down into various intermediates, and once DHAP is formed, that is directly converted to glycerol 3-phosphate by one enzyme called glycerol 3-phosphate dehydrogenase enzyme. So we understand that this enzyme glycerol 3-phosphate dehydrogenase is important to convert the DHAP to glycerol 3-phosphate. So if you talk about this statement, this is a totally right statement, that synthesis is requiring DHAP from glucose, and glycerol 3-phosphate dehydrogenase is playing important role. This is totally true. Now, the other factor I said is the acyl-CoA which is required. Now, the question is from where the acyl-CoA is actually made available in the adipose cell, which is going for esterification and thus storage. The answer are, there are many sources of this acyl-CoA. One is the transfer, the delivery of the acyl-CoA in the adipose cell from the chylomicron and VLDL metabolism. During its metabolism, lot of fatty acid is being delivered to the adipose cell where they are converted to acyl-CoA and then they get esterified for storage purpose. So delivery by this chylomicron and VLDL is one factor, and the other factor is de novo synthesis. Some amount of acyl-CoA is synthesized de novo in the adipose cell for the storage purpose. And the third and important factor is phospholipids. Turnover of phospholipids releases acyl chain, and that is getting esterified and they're getting stored. So the last option which is there in this question, that phosphatidate means phospholipids are hydrolyzed. That's a true statement. So if this question is asking about the wrong statement, and the wrong statement is glycerol kinase, because we do not have glycerol kinase in the adipose cell. Glycerol kinase is found in liver, GIT and kidney. So glycerol kinase is not there in adipose cell. This you remember. So this is going to be the answer in this question.